everybody, and welcome to Bash Course Quarantine Biology. My name is Faith, and today we are going to learn about gene expression. So today's topic is gene expression, but before we can get into that, you need to understand the basics of DNA, RNA, replication, transcription, and translation. So DNA, deoxyribonucleic acid, is a double-stranded nucleic acid made of a backbone of alternating deoxyribose sugars and phosphate groups. Then it has nitrogenous bases, which form hydrogen bonds in complementary base pairing, with adenine binding thymine and guanine binding cytosine. This forms a double helix of base pairs. DNA is the major molecule that stores and passes on genetic information. RNA, ribonucleic acid, is a single-stranded backbone of ribose sugars and phosphate groups. It also has nitrogenous bases, but instead of thymine, it has uracil. It's used as an intermediary in protein synthesis and even has some enzymatic function. DNA replication is accomplished primarily by the enzyme DNA polymerase, which builds complementary daughter strands on each one of the parent strands as they are split by the enzyme helicase. This is called the semi-conservative model of replication, since the new DNA molecule has one original strand and one new strand. The new DNA is built in the 5' to 3' direction. This means that as a replication fork opens up in the original helix, only one strand, called the leading strand, can be built into the direction that helicase is unzipping. The other strand, called the lagging strand, must be made in short segments called Okazaki fragments that start from many primers, primers being RNA sequences attached that tell DNA polymerase where to begin synthesis, and they are laid down by primase. These segments are joined by an enzyme called DNA ligase. Transcription and translation are the steps in protein synthesis to build a protein from the instructions in DNA. Transcription is when an mRNA or messenger RNA molecule is built from a DNA template. This occurs in 3 steps: initiation, elongation, and termination. Initiation is where RNA polymerase attaches to the promoter region of DNA and begins reading down the template strand in the 5' to 3' direction. Elongation is when nucleotides are added to the growing mRNA, and termination is when the mRNA stops being made. The mRNA is then released for processing. Processing only occurs in eukaryotes in the nucleus. Prokaryotes still do transcription, but in the cytoplasm. During processing, non-protein coding regions of RNA, called introns, are spliced out by spliceosomes with snRNAs, small nuclear RNAs, and ribozymes. And exons, protein coding regions, are spliced together. Additionally, a G-cap and poly-A tail of nucleotides are added. Then the mRNA leaves the nucleus and enters the cytoplasm. This is where translation begins. Translation, confusingly enough, also has the 3 steps of initiation, elongation, and termination. The mRNA enters a ribosome, and another type of RNA called tRNA, or transfer RNA, pairs with the nucleotides of the mRNA. This is read in 3 nucleotide sections called codons, which bond with 3 nucleotide anticodons on the tRNA. The ribosome has 3 sites that tRNAs move between: the A, or amino site, P, or peptide site, and E, or exit site. In initiation, the mRNA, large and small subunits of the ribosome, and the initiator tRNA come together, with the tRNA, which codes for methionine, bonding to the start codon AUG. Then, in elongation, the amino acids that are attached to each tRNA are bonded into a polypeptide through peptide bonds as the mRNA is read, and the tRNAs move into the P and then E sites. In termination, a release factor binds to the stop codon and separates the now finished polypeptide from the last tRNA. These processes are how cells with the same DNA can have such different functions. Different genes being transcribed, or in other words, a variable gene expression, is the key. Gene expression is controlled very differently in prokaryotes and eukaryotes. In prokaryotes, operons are used. These are a cluster of genes that are regulated together. There are regulatory genes outside the operon that code for a repressor which can bind to the operator, a regulatory region between the promoter and the operon. And there are 2 main types of operons. An inducible operon is one that usually is switched off. The repressor is bound to the operator, but the presence of a molecule can turn it on. The most well-known example is the lac operon, which switches on when lactose is present so that it can synthesize proteins to break the lactose down. A repressible operon is one that is normally switched on, with a repressor not bound to the operator, but the presence of a molecule activates the repressor and stops gene expression. The main example of this is the trip operon, which controls synthesis of tryptophan. If there is excess tryptophan around, it inactivates the operon because tryptophan synthesis is not needed. That's pretty much it for prokaryotes, but for eukaryotes it's a bit more complicated. The way DNA is packaged around histone proteins affects when it is transcribed. If it's very compact, transcription can be prevented. Another process, called DNA methylation, adds a CH3 group to DNA bases, like cytosine, and makes them less likely to be transcribed. This is only one example of many chemical modifications that can be done to DNA to make it more or less accessible to transcription. These changes can be passed on through what we call epigenetic inheritance, which is inheritance of traits outside of the sequence of nucleotides. Another type of larger scale regulation occurs in women through X inactivation. One X chromosome is modified into a bar body, a highly compact and inactive form. This is passed on through replications, that every cell has an inactive version, but which one is inactive is variable. This is the reason female cats can have a tortoiseshell or calico pattern. It is expressed on the X chromosome, but different areas randomly express either orange or black, depending on which is inactive. On a more microscopic scale, however, eukaryotes use a different mechanism than operons. A gene will have a DNA control sequence called an enhancer, but it will be very far from the gene it regulates. This is where activators will bind, leading to DNA bending and the activators binding the transcription factors. Then the whole complex binds the promoter, and RNA polymerase can attach. Silencers are repressor proteins which can stop this process. Control sequences can be specific, but are often shared with other genes involved in the same metabolic pathways, so all can be transcribed at the same time. Eukaryotic RNA can be edited in different ways through alternative RNA splicing, which allows for the same mRNA to be adapted to different situations by different combinations of exons being used. Eukaryotes also use methods like the carefully timed breakdown of mRNA in the cytoplasm, variable initiation of translation by binding of microRNAs to the mRNA transcript, variable protein activation and protein breakdown to more closely regulate which genes are expressed and when. That's all we have for you on gene expression today. Good luck. Thanks for watching Bash Course Quarantine Biology, and we'll see you next time. The Bash Course Quarantine team is Sandro Ansari, Sam Mencimer, Eamon, Pasha, Ishaq, Sarah Orozco, and myself. Please put any questions you may have in the comments below, and we'll do our best to answer them or get Dr. Green to. Thank you for watching Bash Course, and as they say in my hometown, oh my EOSH.